AFIF, and today I will be your lab assistant. We need to extract the DNA from the E. coli. So first here, we got the E. coli culture. We need to take in 1.5 milliliter of E. coli culture into the centrifuge tube. Okay, but firstly, we need to carry out this process under aseptically condition or under the laminar flow. Firstly, we need to turn on the laminar flow. Okay, then we take the micropipette to pipette the E. coli culture into the centrifuge tube. We take in 1.5 ml of E. coli culture into the centrifuge tube. But first, we take 1000 microliters of this E. coli culture. Okay, next, we set the micropipette to 500 microliters to take the half ml of E. coli culture into the centrifuge tube. Okay, this is the bacterial cell. Next, we need to centrifuge it to differ the cells from the medium, because this is the mixture of the medium and the cell. We use a centrifuge machine and we set up the bacterial cell with a blank sample in a opposite position. This is to avoid the difference in weight, and in long run, it's to avoid the damage to the centrifuge machine. So here we set up at 12,000 RPM for 1 minute. Okay, now you can see here a bacterial culture which have been centrifuged. Here, a two layer is formed. The bottom one is a sediment of bacterial cell particle, or also known as the pellet, and the liquid above the pellet is the supernatant, in which we need to discard. Okay, the liquid we need to discard into the bin. Discard bin. Okay. Next, we take the lysis buffer into our bacterial culture, which has become the pellet on the bottom. We take about 100 microliters of lysis buffer into this centrifuge tube of pellet. This lysis buffer is used to lysing the cell in order to break the bacterial cell to get the DNA of bacteria. Okay, we put the pellet. Next, we need to resuspend with a vortex machine. Okay, that's good. Okay, next we add in 20 microliters of proteinase K into our bacteria. Okay, this proteinase K helps to degrade the protein, and it's also inactivate the nucleases that might degrade our DNA and RNA of the bacteria. Okay, then this we will incubate it at 55 Celsius for 15 minutes. Okay, that's lovely. Okay, after incubation, we take our bacterial cell and then we pipette in to 20 microliters of RNase E into the bacteria. Microliters to our bacteria. This RNase A function is to remove the RNA from the E. coli genomic cell. Then we need to vortex it with a vortex machine. Okay. After vortex, we need to leave it for 2 minutes at room temperature. I'm sorry, I need to go. Raihanah, could you please continue for me?
Hi, I'm Raihanah and I'm going to continue this experiment with you. Let's go! So here you can see the bacteria cell that have been incubated for 2 minutes. So now we are going to add 500 microliters of lysis buffer. Okay, next we need to add the 500 microliters of 96% to 100% of ethanol. So the function of ethanol is to produce the ethanol precipitate, which is in white color. Okay, next is to vortex for 30 seconds for the bacteria cell. Okay, 30 seconds is up. That's all for this step. So next, the bacterial culture must be transferred to the spin column entirely. This is a spin column and is bacteria culture precipitate. So we just transfer them. Okay. Next, the spin column need to be centrifuged for 30 seconds. So here, the centrifuge machine, centrifuge, and need to remember to centrifuge at speed 12,000 RPM. Now, 30 seconds has up. We need to take out the spin column from centrifuge machine, and we need to discard the liquid into the discard bin. So, did you know the difference between spin column and collection tube? So, spin column is used to purify the RNA or DNA for genome isolation. So, the spin column have high salt buffer. So, the DNA will bound selectively to the membrane of the spin column. So, next is the collection tube. So, collection tube is used for DNA purification from blood sample. So, the collection tube mostly have additive serum which will accelerate or prevent blood clotting. Okay. 500 microliters thin buffer 11 was added to the spin column. Okay, after that, we need to centrifuge the spin column. So, always remember, the speed is 12,000 RPM and we need to wait for 30 seconds. 30 seconds was up. So next, we need to take out the spin column from centrifuge machine. So, we need to discard the content into the discard bin. So now, my part are finished. Now Nana, you need to continue for me.
Thank you, Anna. Hello everyone, my name is NURSYAZANA and I will continue with the washing DNA steps. For this step, we will need wash buffer WB11 and also 96% to 100% ethanol. For this washing DNA step, we need to do the step twice, which means after the first time, we need to repeat the same step for another time. Let me show you. Okay, let's start. First, set your micropipette to 500 microliter and add the wash buffer WB11 into your column. And then, using a new tip of micropipette, we also need to add 96% to 100% ethanol into the column. Secure your column. Now, we need to centrifuge this column together with the collection tube for 30 seconds at 12,000 RPM. Now let's wait for 30 seconds. Now our 30 seconds is up. We have to take our column from the centrifuge machine. As you can see here, substance are collected in the collection tube. Discard the flow through into the discard bin. Now, as I said earlier, we have to repeat this step again. Just like earlier, add 500 microliter of wash buffer WB11 into the column and also 96% to 100% ethanol. Again, set your centrifuge machine to 30 seconds at 12,000 RPM. Let the column and also the collection tube to spin at 12,000 RPM for 30 seconds. After 30 seconds, again, we have to take out our column and also collection tube and discard the substance collected in the collection tube into the discard bin. Now, we are done with the washing DNA step. After this, we have to spin the empty column. This is an important step because in the washing step earlier, we used ethanol. We have to spin this empty column because we want to get rid of the excessive ethanol remaining in this column. This is important because in the long run, the ethanol can damage our DNA and we don't want that. So we have to always remember to spin our empty column after the washing step. For this step, you just have to set your centrifuge machine to a point higher than 12,000 RPM for 2 minutes. And then, you just have to let your empty column to spin at the setting you just have set earlier.
After 2 minutes, remove the spin column from centrifuge. Place the spin column into 1.5 ml of microcentrifuge tube. After that, add 100 microliter of sterile distilled water to the center of the column matrix. Incubate at room temperature for 2 minutes. After incubating at room temperature for 2 minutes, place the sample in the microcentrifuge and centrifuge for 1 minute. Remove the tubes from the centrifuge gently. The DNA is now isolated at -20 degrees.